Today, we are diving into a technology that lets us see our own bodies at a level that's just, well, it's almost hard to imagine. We're talking about looking at ourselves one single cell at a time. Let's get right into it.
I mean, just think about this for a second. What if we could look inside a tumor and see exactly which individual cells are resisting treatment? Or what if we could find the very first immune cells that go rogue in an autoimmune disease? This isn't science fiction anymore. This is the incredible promise of single-cell sequencing.
But to really get why this tech is such a huge leap, you gotta understand how we used to do things. And honestly, the best way to think about the old method is to think about a smoothie. Yeah, this brings us to what we can call the cell smoothie problem. You know how it is. You throw in some strawberry, banana, maybe some spinach, and when you taste it, you know all that stuff is in there. But can you pick out an individual strawberry? No way. It's all just blended together into one kind of average flavor. And that is exactly what traditional genetic analysis, what scientists call bulk sequencing, does. It takes a piece of tissue, grinds it up, and then reads the genetic signal of thousands, even millions of cells all at once. So you get the average, but you completely lose all the individual details, all the nuance. So here's the really crucial part. What if the most important cell, that one rogue cancer cell, or that one super important stem cell, is like a single strawberry in a giant smoothie made of spinach? Well, in that averaged out result, its signal is totally drowned out. We'd miss the very thing we were looking for.
So to fix this, scientists realized they had to stop making smoothies and start making fruit salads, right? Where every single piece of fruit is distinct and visible. And to do this, they came up with two really brilliant strategies to analyze each cell one by one.
So the first approach is what's called droplet-based, and it's all about isolation. Think of it like putting each little piece of fruit into its own tiny Tupperware container. The second method is called combinatorial barcoding, and that's all about labeling. It's like giving every single strawberry and blueberry its own unique address label before you even think about mixing them up. Okay, let's break down that droplet method. Using this amazing little device called a microfluidic chip, scientists trap one cell and one special bead inside a teeny tiny droplet of oil. It's basically a personal little lab for each cell. Inside that bubble, the cell pops open, and all its RNA, that's the active genetic messages, grabs onto the bead and gets tagged with a unique barcode. This happens for thousands of cells, and then all the droplets get pooled together. That little barcode on the RNA acts like a return address, telling the scientists exactly which cell it came from. So cool. Now, the combinatorial method is just as clever, but it uses a totally different kind of logic. Here, cells start out in a plate with lots of little wells, and everything in a single well gets the same first barcode. Then, and this is the fun part, all the cells are pooled together, mixed up, and then split back out into a new plate to get a second barcode. You do this a few times, and each cell ends up with a completely unique combination of barcodes. It's like a multi-part zip code that pinpoints its identity. Okay, so we have these two incredible strategies, but, how do they actually stack up in the real world? Well, a recent study in the International Journal of Molecular Sciences did a direct head-to-head comparison of two of the leading platforms that use these very different approaches. So first, let's look at efficiency. This is basically asking how many of the cells you start with do you actually get data from? And as you could see, the droplet-based platform from 10x Genomics, it recovered about 53% of its cells. Pretty good. The combinatorial platform from Parse Biosciences, on the other hand, only got about 27%. So just looking at this one number, you'd say, okay, clear win for the droplet method.
But wait, that is not the whole story. Because when you look at sensitivity, which is how many genes are you detecting inside each cell, the whole picture flips. Check this out. The combinatorial platform from Parse detected over 2,300 genes per cell, while the droplet one detected just over 1,900. So yeah, you get fewer cells back, but you get a much, much deeper look inside each one.
And this quote from the researchers just nails why that sensitivity is so important. They basically said that because the Parse platform detected more genes, it was actually able to find rare cell types that the other platform missed completely. Remember that single strawberry in the smoothie? Higher sensitivity is how you find it.
So what we have here is a classic scientific trade-off, right? There's no single best answer. 10x genomics gives you higher efficiency, so you get data from more of your cells. But parse biosciences gives you that higher sensitivity, letting you see more genes, and this is a big one, it can handle way more samples at the same time, which is a huge deal for big research projects.
All right, so you've done the experiment, you've gotten all this sequence data, but that's really only half the battle. The next huge challenge is, what do you do with it all? I mean, the raw data that comes out of one of these experiments is just staggering. Seriously. Imagine a giant spreadsheet with thousands of columns, one for every single cell, and tens of thousands of rows, one for every gene. It's just a sea of numbers. So the big question is, how do you find the hidden patterns in all that? How do you turn all that noise into a real, meaningful biological story? Well, the answer is a field called bioinformatics. Scientists use incredibly powerful software. There's a great open source tool called Scanpy, for example, to make sense of this data ocean. These computer programs act like a super smart sorting machine, automatically grouping cells that have similar genetic activity. And just like that, it separates out all the different cell types, your T cells, your B cells, your fibroblasts, and reveals the true diversity that was hiding in that tissue sample all along. Now, this technology is already mind-blowing, but the field is moving at an absolutely blistering pace. And the next huge mountain to climb is all about three things: location, location, location. Think about it for a second. Both of the methods we talked about, the droplets and the barcoding, they both require you to break the tissue apart into individual cells right at the very beginning. So we learn who all the cells are in amazing detail, but we completely lose the information of where they were. Who were their neighbors? What was the local environment like? That's all gone. So we've taken our beautiful fruit salad and turned it into just a list of ingredients. The next great frontier is figuring out how to put that map back together, to link every cell's unique identity back to its original physical address in the tissue. And this emerging field has a name. It's called spatial transcriptomics. These new technologies are being developed to analyze the genes of a cell while it is still in place within a slice of tissue. The goal is to create a true high-resolution cellular map, showing not just what the cells are, but how they're all organized and talking to each other. Which leads us to a final incredible thought. As we get closer to building a complete atlas of the human body, knowing every single cell type and exactly where it lives, what becomes possible? I mean, from decoding how the brain develops to designing cancer therapies that are targeted with pinpoint accuracy, the potential is just transformative. It really just leaves you wondering, where are we going to see the biggest revolution first?